Vaccine and antibody. Vaccine. Vaccine is a biological agent, bacteria, fungus, virus, etc., that elicit an immune response to a specific antigen derived from an infectious disease-causing pathogen. Generally, it comprises an agent that has a resemblance to the disease-causing microbe. Immunization. It is the action of making a person or animal immune to infection, typically by inoculation. Types of vaccine. 3 main approaches to making a vaccine. 1. Using a whole virus or bacterium. This is the traditional approach to vaccine development, and it is still used for many vaccines today. In this approach, the virus or bacterium is grown in a laboratory and then weakened or killed. The weakened or killed virus or bacterium is then injected into the body, where it triggers an immune response. Examples of vaccines that are made using this approach include measles, mumps, and rubella, MMR, vaccine, polio vaccine, chickenpox vaccine, rabies vaccine. Types of vaccines that use a whole virus or bacterium. 1. Inactivated vaccines. Inactivated vaccines contain whole virus particles that have been killed or inactivated so that they can no longer cause disease. These vaccines are generally very safe and effective, but they may require multiple doses to achieve full protection. 3. Examples of inactivated vaccines include flu, polio vaccine, Covaxin. 2. Live attenuated vaccines. Live attenuated vaccines contain weakened live virus particles that can still replicate in the body, but they are not strong enough to cause disease. These vaccines are generally very effective at providing long-lasting immunity, but they may have more side effects than inactivated vaccines. They are also not suitable for people with weakened immune systems. Examples of live attenuated vaccines include measles, mumps, and rubella, MMR, vaccine, chickenpox vaccine, yellow fever vaccine, rotavirus vaccine. 3. Virus vector vaccine. Virus vector vaccines use a harmless virus to deliver genetic material from the target virus or bacterium into the body. This triggers an immune response to the target virus or bacterium without the need to inject the whole virus or bacterium into the body. Virus vector vaccines are a newer type of vaccine, but they have shown great promise in clinical trials. They are particularly well suited for developing vaccines against emerging diseases, such as COVID-19. Examples of virus vector vaccines include Ebola vaccine, Covishield. 4. 2. Using parts of the virus or bacterium that trigger the immune system. This approach is becoming more common, as it allows scientists to develop vaccines that are more targeted and less likely to cause side effects. In this approach, scientists identify the parts of the virus or bacterium that are most important for triggering an immune response. These parts of the virus or bacterium are then produced and used to make the vaccine. Examples of vaccines that are made using this approach include whooping cough, tetanus, diphtheria. 3. Using just the genetic material of the virus or bacterium. This is the newest and most advanced approach to vaccine development. In this approach, scientists use the genetic material of the virus or bacterium to create a vaccine. This is done by inserting the genetic material into a harmless virus or bacterium. When this harmless virus or bacterium is injected into the body, it triggers an immune response to the virus or bacterium that is being targeted. Examples of vaccines that are made using this approach include mRNA vaccines for COVID-19, DNA vaccines for COVID-19. Other approaches to making a vaccine. Toxoid vaccine. A toxoid vaccine is made by inactivating the toxin that causes disease symptoms. The toxin is a poisonous substance produced by bacteria or viruses. When the toxin is inactivated, it is no longer able to cause disease, but it can still trigger an immune response. Toxoid vaccines are very effective at preventing diseases caused by toxins. Examples of toxoid vaccines include diphtheria vaccine, tetanus vaccine, pertussis vaccine, the T in DTaP and Tdap. Conjugate vaccine. A conjugate vaccine is made using a combination of 2 different components, polysaccharide and a protein carrier. A polysaccharide is a type of sugar molecule that is found on the surface of many bacteria. A protein carrier is a protein that is very good at triggering an immune response. Polysaccharides on their own are not very good at triggering an immune response in young children. However, when they are conjugated to a protein carrier, they become much more immunogenic. Conjugate vaccines are very effective at preventing diseases caused by bacteria that have polysaccharides on their surface. 5. Examples of conjugate vaccines include Haemophilus influenza type B, HIB, vaccine. Pneumococcal conjugate vaccine, PCV13. Meningococcal conjugate vaccine, Menaqui. Inactivated vaccine working mechanism. Inactivated vaccines are made from viruses or bacteria that have been killed or inactivated so that they can no longer cause disease. When an inactivated vaccine is injected into the body, the immune system recognizes the virus or bacterium as a foreign invader and mounts an immune response. The immune response produces antibodies, which are proteins that can bind to the virus or bacterium and prevent it from infecting cells. If the person is later exposed to the virus or bacterium, the antibodies will recognize and neutralize it, preventing the person from getting sick. A step-by-step explanation of how an inactivated vaccine works. 1. The vaccine is injected into the muscle. 2. Dendritic cells, a type of white blood cell, take up the vaccine particles and transport them to the lymph nodes. 3. In the lymph nodes, the dendritic cells present the vaccine particles to T cells, another type of white blood cell. 4. Some of the T cells become activated and differentiate into helper T cells and killer T cells. 5. The helper T cells release cytokines, which are signaling molecules that help to activate other immune cells, including B cells. 6. B cells divide and produce antibodies, which are proteins that can bind to the virus or bacterium and prevent it from infecting cells. 7. Some of the B cells become memory B cells, which can quickly produce antibodies if the person is later exposed to the virus or bacterium. Inactivated vaccines are very safe and effective. They are generally well tolerated, with few side effects. Inactivated vaccines are also very effective at preventing disease. However, they may require multiple doses to achieve full protection. Some examples of inactivated vaccines include flu vaccine, polio vaccine, hepatitis A vaccine, rabies vaccine, Covaxin. 6. Viral vector vaccine working mechanism. Viral vector vaccines use a harmless virus to deliver genetic material from the target virus or bacterium into the body. This triggers an immune response to the target virus or bacterium without the need to inject the whole virus or bacterium into the body. To make a viral vector vaccine, scientists first modify a harmless virus to remove its ability to cause disease. Then, they insert genetic material from the target virus or bacterium into the modified virus. When the modified virus is injected into the body, it infects cells and delivers the genetic material from the target virus or bacterium into the cells. The cells then produce proteins from the target virus or bacterium. These proteins are then displayed on the surface of the cells, where they are recognized by the immune system. The immune system then mounts an immune response to the proteins, producing antibodies and T cells that can protect the body from the target virus or bacterium. A step-by-step explanation of how a viral vector vaccine works. 1. The vaccine is injected into the muscle. 2. The vaccine particles are taken up by cells in the muscle. 3. The vaccine particles enter the nucleus of the cells. 4. The genetic material from the target virus or bacterium is transcribed into mRNA. 5. The mRNA is translated into proteins. 6. The proteins are transported to the surface of the cells and displayed. 7. The proteins are recognized by the immune system. 8. The immune system mounts an immune response to the proteins, producing antibodies and T cells that can protect the body from the target virus or bacterium. Viral vector vaccines are a newer type of vaccine, but they have shown great promise in clinical trials. They are particularly well suited for developing vaccines against emerging diseases, such as COVID-19. 7. Some examples of viral vector vaccines include AstraZeneca COVID-19 vaccine, Johnson & Johnson COVID-19 vaccine, Covishield. Primary and secondary immune response, naturally. Primary immune response. The primary immune response is the first time the body encounters a new antigen. An antigen is a substance that is recognized by the immune system as foreign. When the body encounters a new antigen, the immune system begins to produce antibodies against it. The primary immune response is typically slower and weaker than the secondary immune response. It can take several days or even weeks for the body to produce enough antibodies to protect against the antigen. Secondary immune response. The secondary immune response is the body's second response to an antigen. If the body is exposed to the same antigen again, the immune system will be able to respond more quickly and more strongly. This is because the body has already created memory B cells against the antigen. Memory B cells are a type of B cell that remains in the body after the primary immune response has subsided. If the body is exposed to the same antigen again, the memory B cells can quickly produce antibodies against it. This helps to protect the body from getting sick again. The image you sent shows how the primary and secondary immune responses differ in terms of speed and strength. The primary immune response is slower and weaker, while the secondary immune response is faster and stronger. Example of primary and secondary immune response. Imagine that you are exposed to the measles virus for the first time. Your body will mount a primary immune response to the virus. This means that your immune system will begin to produce antibodies against the virus. It may take several days or even weeks for your body to produce enough antibodies to protect you from the virus. 8. If you are exposed to the measles virus again after you have recovered from the first infection, your body will mount a secondary immune response. This means that your body will be able to respond more quickly and more strongly to the virus. This is because your body has already created memory B cells against the virus. The memory B cells will quickly produce antibodies against the virus, which will help to protect you from getting sick again. Primary and secondary immune responses are essential for protecting the body from infectious diseases. Vaccines work by triggering a primary immune response to an antigen. This helps to prepare the body to fight off the infection if it is exposed to the antigen again in the future. Primary and secondary immune response, vaccine. Primary immune response. The primary immune response to a vaccine is the body's first response to the vaccine antigens. It is initiated by dendritic cells, which are antigen-presenting cells. Dendritic cells take up the vaccine antigens and transport them to the lymph nodes, where they present them to T cells. T cells are a type of white blood cell that play a central role in the immune response. T cells can be divided into two main types, helper T cells and killer T cells. Helper T cells help to activate other immune cells, including B cells. Killer T cells directly attack infected cells. When a helper T cell is activated by a dendritic cell presenting a vaccine antigen, it releases cytokines. Cytokines are signaling molecules that help to regulate the immune response. The cytokines released by helper T cells activate B cells and promote the differentiation into antibody-producing plasma cells and memory B cells. Plasma cells produce and secrete antibodies, which are proteins that bind to specific antigens. Antibodies can neutralize antigens, preventing them from infecting cells. Memory B cells remain in the body after the primary immune response has subsided. They can quickly produce antibodies if the body is exposed to the same antigen again. The primary immune response to a vaccine typically takes several weeks to develop. During this time, the body is not fully protected from the infection that the vaccine is designed to prevent. However, the primary immune response primes the immune system to respond more quickly and more strongly to the infection if the body is exposed to it again in the future. Secondary immune response. The secondary immune response to a vaccine is the body's second response to the vaccine antigens. It is initiated by memory B cells. When a memory B cell encounters a vaccine antigen, it quickly divides and produces antibodies against the antigen. The secondary immune response is much faster and stronger than the primary immune response. This is because the memory B cells are already programmed to produce antibodies against the vaccine antigens. The secondary immune response typically develops within a few days of exposure to the vaccine antigens. 9. Importance of primary and secondary immune responses to vaccines. The primary and secondary immune responses to vaccines are essential for protecting the body from infectious diseases. Vaccines work by triggering a primary immune response to the vaccine antigens. This helps to prepare the body to fight off the infection if it is exposed to the antigen again in the future. The secondary immune response is responsible for the long-lasting immunity that is provided by vaccines. When the body is exposed to the vaccine antigens again, the memory B cells quickly produce antibodies against the antigen, preventing the infection from developing. Booster shots. Booster shots are given to maintain the body's immunity to a particular infection. Booster shots are typically given several years after the primary vaccination series. They are important because levels of antibodies in the blood can decline over time. Booster shots help to ensure that the body has a high enough level of antibodies to protect against infection. Antibody. An antibody, also known as an immunoglobulins, Ig. It is Y-shaped protein. It helps to identify and neutralize foreign objects such as pathogenic bacteria and viruses. Antigens. Antibodies are Y-shaped proteins that are produced by B cells in response to infection. They bind to specific antigens, foreign invaders, and help to neutralize them. 10. IgA. IgA is the most common type of antibody in the body. It is found in the blood, saliva, tears, and breast milk. IgA protects the body from infection by preventing pathogens from attaching to the surfaces of cells. IgD. IgD is found on the surface of B cells, which are a type of white blood cell. IgD helps to activate B cells when they encounter a pathogen. IgA. IgA is involved in allergic reactions. It binds to mast cells and basophils, which are types of white blood cells that release histamine and other chemicals during an allergic reaction. Histamine and other chemicals cause the symptoms of an allergic reaction, such as sneezing, runny nose, itchy eyes, and hives. Ig. Ig is the most abundant type of antibody in the blood. It is responsible for long-term immunity to infection. Ig antibodies can cross the placenta, which means that they can protect a baby from infection before and after birth. IgM. IgM is the first type of antibody that the body produces in response infection. Antibodies to IgM are very effective at neutralizing pathogens, but they are not very good at activating other immune cells. Plasma convalescent therapy. Plasma convalescent therapy is a type of passive immunotherapy that uses plasma from people who have recovered from COVID-19 to treat people who are currently infected with the virus. Plasma is the liquid part of blood that contains antibodies and other proteins that help the body fight infection. Antibodies are proteins that bind to specific antigens, which are substances that trigger an immune response. When a person is infected with COVID-19, their body produces antibodies against the virus. These antibodies can help to neutralize the virus and prevent it from infecting cells. Plasma convalescent therapy works by providing patients with antibodies from people who have already recovered from COVID-19. This can help to boost the patient's immune system and fight off the infection. Plasma convalescent therapy is still under investigation, but it has shown some promise in clinical trials. In one study, plasma convalescent therapy was found to reduce the risk of death by 35% in hospitalized patients with COVID-19. 11. Plasma convalescent therapy is typically given to patients with severe or life-threatening COVID-19. It is also sometimes given to patients who are at high risk of severe complications from COVID-19, such as older adults and people with underlying medical conditions. To administer plasma convalescent therapy, plasma is collected from a donor who has recovered from COVID-19. The plasma is then screened to ensure that it is safe and that it contains high levels of antibodies against the virus. The plasma is then transfused into the patient. Plasma convalescent therapy is generally safe and well tolerated. However, there are some potential side effects, such as allergic reactions and fluid overload. Plasma convalescent therapy is one of a number of treatments that are being investigated for COVID-19. It is an important tool for treating people with severe or life-threatening COVID-19, but it is important to note that it is still under investigation and more research is needed to determine its Safety and efficacy. The image shows how plasma convalescent therapy works to treat COVID-19. The image shows a COVID-19 patient who is receiving a transfusion of plasma from a donor who has recovered from COVID-19. The plasma contains antibodies against the SARS-CoV-2 virus, which causes COVID-19. The antibodies bind to the virus and neutralize it, preventing it from infecting cells. Q. With reference to recent developments regarding recombinant vector vaccines, consider the following statements. 2021. 1. Genetic engineering is applied in the development of these vaccines. 2. Bacteria and viruses are used as vectors. Which of the statements given above is correct? A. 1 only. B. 2 only. C.